Rahim. So now that we have the gonad, we'll discuss how these gonads are going to produce their gametes. Excuse me. Now, the gametogenesis, or the process of producing the gametes, is known as gametogenesis, and this occurs through a process known as meiosis. And meiosis is a two cell division process. First, there is first meiotic division, and then there is the second meiotic division. So, in order to discuss that, we must see generally what happens is this is one cell, and it goes through the first meiotic division, and it forms two cells right here. And then these two cells will undergo the second meiotic division, and each of the two cells will again divide and produce two cells, so a total of four cells are produced, as you can see. So we started as one cell right over here, and now we end up with four cells at the end of the second meiotic division. So we'll have to discuss it a few times because there are differences between the male, what in meiosis between male and females, but there are some similarities as well. First, we'll discuss the similarities between the male and female meiotic process. So, what happens in the gonads is this type of cell is present, and it is called in spermatogonia or a oogonia. A spermatogonia oogonia. This is like a mitotic cell. This is like any other somatic cell that replicates itself through a process of mitosis. It just makes more copies of itself. So this is the spermatogonia or oogonia, and this is just like another somatic cell. All somatic cells, they undergo the mitotic process, not the meiotic process. But some of these cells, they become meiotically active, and they're committed to undergo meiosis. So when these cells become committed to meiosis, they are known as the primary spermatocyte or the primary oocyte. So these cells will only undergo meiosis, they will not undergo mitosis. So the first step in the meiotic process or the mitotic process, in any case, any type of cell division, is that the DNA has to replicate itself. As you can see that we started with a pair of chromosomes, and you know that there are 23 pairs of chromosomes. This is one chromosome and this is another chromosome. So this is a pair of chromosomes and the number of chromosomes, there are 23 pairs of chromosomes, so the number is 46. The total number of 46 chromosomes, and it's a diploid number. Excuse me. Or 2n. The simplest way of remembering this is that consider one chromosome as 1n. Consider one chromosome as 1n. So since there is a pair of chromosomes, so consider it to be 2n or diploid. So there are 23 sets of these 2n chromosomes. It makes 46. So 46 chromosomes, a number is diploid, which is 2n. So since the DNA has replicated itself, so there is now twice as much DNA in this cell as compared to this cell, or the primary spermatocyte or the primary oocyte. The number of chromosomes remain the same. It's the same two chromosomes or a pair of chromosomes. This is one chromosome and this is the other chromosome. But the DNA has doubled. The DNA has doubled, so it becomes 4n. So now there are four strands of chromosomes. Each strand of chromosome is known as a chromatid. So this is one chromatid and this is the other chromatid, and they're held together at the center by a point known as the centromere. So we've gone from 46 chromosomes with a 2n or diploid number to 46 chromosomes with a 4n or tetraploid number. The DNA has doubled itself. Now we have four strands or 4n number of chromosomes, but the number of chromosomes remain the same, that is 46. So now this pair of chromosomes, they undergo a cell division, which is the first meiotic division, and each of these pairs of chromosomes, they go into another, each daughter cell. This pair, this this chromosome goes into this daughter cell, and this chromosome goes into this daughter cell. So each member of this pair of chromosomes separates and goes into each daughter cell. So now each daughter cell will have 23 chromosomes instead of 46. And the number becomes diploid again. From tetraploid, it becomes diploid again. So it's 2n. So there are two strands of chromosomes, or you can say that they're two chromatids held together by the centromere. So we started from a single cell, which is a somatic cell, and a spermatogonia or oogonia. And we went from a single cell that was diploid, 46 chromosome diploid number, to a committed cell, meiotically active, to 46 with 46 chromosomes and tetraploid number. And now, after the first meiotic division, we end up with a cell which is containing 23 chromosomes with a diploid number. So now these two daughter cells will undergo the second meiotic division, and as you can see, that what happens in the second meiotic division is this is centromere, it splits. The centromere, it splits, and the chromatids, they separate, and each chromatid or each strand of DNA goes into a separate cell. As you can see, this one chromatid goes into this cell, the other chromatid going into the other cell. And similarly, this is what happens in the second daughter cell. So now the number of chromosomes remain the same. It's the same 23 chromosomes, but the DNA, the amount of DNA has gone from 2n to 1n. The amount of DNA is further reduced to 1/2 of what we had here. So now this 1n number is called a haploid number. So this is, let's say we've gone from a cell which had 46 chromosomes with a diploid number to 23 chromosomes with a haploid number. So this is what happens both in the male and the female. This is what is similar between the male and the female. In the next video, we'll discuss the differences.